Hey, hi everyone. Uh, it's Rethik Sir, your biology SME, and welcome back to the biology series. So, in the last few videos, we actually talked about what? Principle of inheritance, and we have also touched some of the basis of mutation. So, not only that, even pedigree analysis. So, now what's mutation is all about? Well, it's when a change happens in the location of gene or when a change happens in our DNA, which can lead to what? A different traits or different characters, and sometimes even diseases, like a kind of disorders. Sounds interesting, right? Yeah. Today we will get even deeper into what exactly the mutation is and what kind of disorders, okay, that comes when there is a mutation happens in genomic level as well as in a chromosome level. So first thing is first, what exactly the mutation is? A mutation is a change in a DNA sequence. So this change can affect the organism's genotype. Genotype is which it is a genetic makeup, and a phenotype. Phenotype is a physical trait that we normally see, right? So thinking about the genetic disorder, so there are two types that we come across. So genetic disorder may be grouped into two categories. One is Mendelian disorder, and the second one is the chromosomal disorder. So what is Mendelian disorder? It has always seems impossible until it's done. It's nothing but if you want to do something, it might feel impossible, but unless until it's done. If you want to complete a chapter today, so it might feel impossible. Oh, time is not there, or it is somewhat tricky, tough. I am unable to remember. But unless you try, you won't get to know whether it is really impossible or really it is so tough that the way you are imagining, or it's just a myth. So unless until you try, to not come to a conclusion saying that it is really impossible. Mendelian's disorder. A main when I'm talking about a Mendelian disorder, they are mainly determined by an alternation or a mutation in a single gene. So, if it is a DNA, say like if it is a DNA, a single stranded DNA, so genes are present on the DNA. So, if there is any mutation or alternation that has happened in a single gene, okay, in a single gene, so it could be either by the sublimation or unknowingly if the gene has been a new gene has been inserted or a gene has been deleted from its location, so this might leads to what? Disorder or some kind of variation. So now a DNA, it has got mutated or a gene got mutated or a gene has changed its location, something happened now, okay? So these disorders, and now it created a disorder. So now these disorders, my friends, they are transmitted to the offspring on the same line. Same line matlab, if you have remembered the Mendelian principle, you exactly know how the traits have been carried from one generation to the another generation, say like from parents to F1 generation and from F1 generation to next generation, that is F2 generation. In the same way, these disorders which have been created, right, so they will be transmitted to the offspring on the same line. So the pattern of inheritance of such Mendelian disorder can be tracked by like in a family by using what? Pedigree analysis. By using pedigree analysis. Clear, my friends? So is it really clear to you? So what exactly the Mendelian disorder is? Nice. So some of the best examples of Mendelian disorders are most common example. One is hemophilia, then the cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia are some of the examples regarding Mendelian's disorder. So now we will dig in detail regarding each examples. So Mendelian disorders, whenever I'm saying it could be dominant or recessive, just like we have studied in the principle of inheritance. So by using this pedigree analysis, one can easily understand whether the trait is in a question, that is whether the trait is dominant or whether it is recessive. that is the trait which is moving from one generation to another generation, whether the trait is dominant or it is recessive. So how one can actually easily understand it by analyzing the pedigree. The trait may also be linked to the sex chromosomes. Many of the cases, say like an example, hemophilia. So in hemophilia, what happens? It is evicted that X-linked recessive trait shows a transmission from carrier female to male progeny. So here what is happening? So it is showing a trait is actually showing a transmission from carrier female to the male progeny. And this is a chart of a pedigree showing autosomal dominant trait and autosomal recessive trait. So in autosomal dominant trait, what happens? So any one parent character, that is any one parent's trait is going to carry to their offspring. Whereas in autosomal recessive trait, both the parents' character both the parents' trait is going to transfer or it is going to trans it in their offspring. Okay. So this is a simple definition or simple observation that we can do regarding this. So here in the figure, you can see in autosomal dominant traits, so only mother is affected. That's what the figure is showing. And each offspring of the generation has been affected. So here you are not seeing any complete affected kind of symptoms, right? But you can see that in the next generation, all their offsprings, so they are being affected. So that means here, obviously, the trait must be inherited by both the parents. Here, trait is actually inherited by any one of the parent. So that is the differences between the autosomal dominant and autosomal recessive trait. So coming to the very first disorder of Mendelian's disorder, that is color blindness. So color blindness, it is a sex-linked recessive disorder, and this is due to the fact that the the cone of the eyes is unable to discriminate between the red and green color. Okay. So the cone of the eyes resulting in failure to discriminate between the red and green color will leads to what? Color blindness. So it occurs in 8% of the male and only 0.4% in the female. This is because, you know, the gene which leads to red-green color blindness are actually present on the X chromosome. So now we know that the male will be having only one X chromosome and the other one is Y chromosome, where female will be having two. So the son of the woman who carries the gene has 50% chances of being color blindness and another 15% we don't know. Okay. So chances of becoming a color blindness is 50%. This is what they're trying to say. So the mother is not herself colorblind because what happens, as I told here, the gene is recessive. So that means that the effect will be suppressed by her matching dominant normal gene. So if a affected, that is carrier mother, is actually married with the normal father, as a result, what happens? So in the next generation, the daughter will be carrier or a normal daughter. But out of the son, the chances of expression is 50%. That means, so one among two sons, they may get this color blindness. That is what the pedigree analysis says. So if a daughter will have to become a color blind, normally at this circumstance, a daughter will be a carrier. Okay, but if a daughter will normally be a color blind, unless until if the mother, who is actually a carrier, right, she is married with a father who is color blind. So now what happens as a result? A father is already a color blind. That means the X chromosome, obviously, it is also going to carry this thing. What do you say that? It is also going to carry this color blindness gene. And even a mother also is going to carry this color blindness gene. So as a result, all the son will be color blind and the daughter might also become a color blind. So this is a proportional ratio. But normally what happens, since a mother is a carrier, so the expression rate is more in the male when compared to the female. This is clear. Going to the next topic, that is hemophilia. Again, in hemophilia, as in the figure you can see the normal blood vessels, so if there is any damage happened, so it gets clotted. But in the hemophilia, what happens, the blood vessels, so when there is any clotting happens, so now this normal blood vessels, it is unable to get recovery, it is unable to get clot, so it loses its clotting capacity. Let us see it in depth. As I told, it is also a sex-linked recessive disease. It shows its transmissions from unaffected carrier female to some of the male progeny has been widely studied. A sing In the hemophilia, what happens? A single protein, which is actually a part of cascade cascades of proteins, so that normally involved in the clotting of the blood, has actually been affected. Okay. So, in an affected individual, what happens as a result? Even a simple or minute cut will result in the non-stop bleeding. So, a single protein that is actually a part of cascade of proteins, okay, that involves in the clotting of the blood. So, that protein has been affected. So, the protein which is actually involved in the clotting of the blood has been affected. So, if that has been affected, as a result, what happens? So, that leads to non-stop bleeding if some a small, minute, cut has also happened. So, if a female is a heterozygous, so what do you mean by heterozygous? So, this could be an heterozygous, right? So, if a female is an heterozygous, so for hemophilia, may transmit the diseases to son. Clear? So, almost everywhere, the mother will be the carrier here in recessive. So, what happens in an heterozygous female, that is the carrier female, the hemophilia may transmit the disease to her son. So, the possibility of the female becoming an hemophilic is again extremely rare, just like I discussed in the previous one. Because here also, mother of such female has to be at least a carrier and the father should be hemophilic. Okay. Same like the previous which I have discussed. Even here also, the female becoming an hemophilic is extremely rare because a mother of such female, has to be at least a carrier and the father should be hemophilic. So if this case happen, then the possibility of the female becoming a hemophilic is possible. So one best example, the hemophilia was being studied is in the family pedigree of the Queen Victoria, which actually shows a number of hemophilic descendants as she was the one who carrier of the disease. Hence, we also call it as royal disease. Okay. So because it was very first studied in detail in the family pedigree of the Queen Victoria. So moving to the next disorder, that is sickle cell anemia. So I want you to watch this graph or watch the small video to know how the shape has been changing. That is the RBC blood cells shape. It gets changing. Yes. Again, this sickle cell anemia is again an autosome linked recessive trait that can be transmitted from the parents to the offspring when both the parents are carriers for the gene. Here, what happens? We need to understand this is somewhat different. Here, it is again an autosomal linked recessive trait, but that can be transmitted from the parents to the offspring only when both the parents are carrier of the gene. That is, when both the parents, that is during the heterozygous condition. We will see how and why. Now, the disease is actually being controlled by a single pair of allele. So HBA and HBS. So as a result, what happens? Out of three possible genotype, only homozygous individual, you're getting, only homozygous individual is going to get affected, or it is going to show this disease phenotype. That is homozygous condition in sense if it is HBS and HBS condition. So is it homozygous condition? So three possibilities is one is HBA and HBA. This is one possibility. Then HBA and HBS is another possibility, and HBS and HBS. So this is one possibility. Out of these three genotype possibilities, only homozygous individual, which is HBS and HBS, right? So it is going to show this disease phenotype. And I told the disease is controlled by single pair of allele. So this is what I'm trying to say. So in heterozygous individual, what happens? So if it is in this condition, if it is in this condition, what happens? So they will be unaffected, but now this will be the carrier. Okay, so they will be unaffected, but they will be the carrier of the disease, as there is 50% of probability of transmission of this mutant gene to the progeny. Thus exhibit what? Sickle cell trait. So this is clear to you. So sickle cell anemia, it shows its all symptoms and all only at homozygous condition. At heterozygous condition, what happens? It behaves as a carrier. So it might get carried. Okay. The one who is affected, they will act as a carrier. So this is clear. So one mechanism that I really want to, say how this happens, how this mutation actually happens. So are you curious to know how the sickle cell anemia mutation normally happens, right? See, what happens is the defect is actually caused by the substitution of this glutamin, of this glutamic acid. So what happens? The glutamic acid that you are seeing here, right, which is actually in the sixth position, that gets substituted by this, that is valine. Okay, so the glutamic acid gets, which is actually present in the sixth position. Okay, of what? Of beta globin chain. Okay, so the glutamic acid, which is actually present in the sixth position of the beta globin chain of the hemoglobin molecule, so it gets what? Substituted, okay, by valine. Okay, substituent of this amino acid sequence you're seeing. So by what? Valine. So as a result, what happens? As a result, what happens? This is substitution of this amino acid in the globin protein resulting due to the single base substitution at the sixth codon. So can I call it as a codon here? So this is a codon. Obviously, if there is some kind of, uh, position. So what happens there? Even the codons also gets changed. So instead of what happens here, so the change that you can see here is this beta globulin gene. Okay, beta globulin gene from the CAC, it gets substitutes or changed to GUG. Okay. As a result, what happens? The mutant hemoglobin molecules undergo the polymerization under low oxygen tension. So this actually undergoes what? Polymerization under low oxygen tension. Okay, causing the change in the shape of the RBC. So what is the shape of the RBC? That is biconcave shape. So as I have shown in the previous video, how the shape got changed, it got changed into elongated sickle-like structure. Okay, so this is clear. So what is the mechanism? So one thing is in the substitution that actually happens of the, what do you say, that glutamic acid, so in which is actually present in the sixth position of the beta globin chain of the hemoglobin molecules, the get substituted by valine. Okay. So as a result, what happened? The substitution of the amino acid in the globin protein resulting due to the single base substitution at the sixth codon of the beta globin gene from CAC to GUG. As a result, what happened? The mutant hemoglobin molecule, they undergo this polymerization under low oxygen tension, which results in the change of the structure or shape of the RBC from biconcave disc to elongated sickle-like structure. So this is all about sickle cell anemia and how it actually happens. The next disease is thalassemia. So it is also an autosomal linked recessive blood disease which is transmitted from the parents to the offspring. When both the parents are unaffected carrier of the gene, that means both the parents are not affected, but they are carrier of the gene. Okay, during heterozygous condition. So this is commonly seen. So the defect could be due to either mutation or deletion, which ultimately result in the reduced rate of synthesis of one of the globulin chain. See what happens in that makeup of hemoglobin. So in the hemoglobin, so we normally see two chains. One is alpha and beta chain that normally make up the hemoglobin. So if there is any mutation that happened in any of this chain, or if there is any deletion that happened, ultimately it leads to the reduced rate of synthesis of one of the globulin chain, okay, that make up the hemoglobin, and thalassemia has been observed now. So this causes the formation of what? An abnormal hemoglobin molecules, okay, resulting into this is called anemia, which is characteristic of the disease. So thalassemia can be classified into two, as I said, based on which molecule it has affected. One is alpha and beta thalassemia. So in alpha thalassemia, production of this alpha globulin chain has been affected, whereas in beta thalassemia, production of beta globulin chain is obviously affected. It's very common sense. So in the alpha thalassemia, it is actually controlled by two closely linked genes. So one is HBA1 and HBA2 are the gene which is closely linked, which normally locates in the 16th position of the chromosome, okay, of each parent. And it is observed due to either mutation or deletion in one or more of these four genes. Okay, so as a result, what happens? The mutation might occur. That means, see, in one parent, you will see these two genes. So now I'm talking about two parents, totally four genes. So out of this four gene, if any one of the gene is been deleted or mutated, or more of these four genes have been deleted or mutated, so alpha thalassemia might occur. So what happens? The more genes affected means the less alpha globin molecules will be produced. This is simple. So while in beta thalassemia, it is controlled by what now? Single gene on both the parents. They are actually present on the 11th position of the chromosome in each parents. And again, this also is occurred due to mutation or deletion of any one or both the gene. So in one parent, you will see HBB. So in both the parents, you will see two HBB, HBB from mother and another from parent. So if any one of the gene has been mutated or deleted, or both the gene has been mutated or deleted, will leads to what? Beta thalassemia. So this thalassemia differs from the sickle cell anemia. Okay. How? Because there also I was talking about beta globulin chain. Here also I'm talking about beta globulin chain. But it completely gets differs from their sickle cell anemia, because what happens here, the basically what happens, it actually forms a quantitative problem of synthesizing to few globulin molecules. Okay, that means what happens if a person has been affected by the beta thalassemia, as a result what happens, so starting a very few, okay, very few globulin molecules will be synthesized, but later what happens, the problem is it starts synthesizing an incorrect functioning globulin. Okay. As a result, what happens? So if it starts synthesizing an incorrect functioning globulin, so body disorders could be occurred, or that symptoms will be noticed, will be seen. So this is all about what? Thalassemia. Okay. And in general, it was all about a Mendelian disorder. So what happens if a gene position has been changed? So we have studied about what happens if a gene has been mutated or gene has been deleted. So as a result, what could be the disorder and how, in which position if they gets mutated. So one thing is clear, like I was discussing, it is present in the 12th position of the chromosome, 11th position of the chromosome. So one thing is clear, that this gene has its own specific position on the chromosome. So if that gene has been disturbed from its position, if they are being mutated or if they are being deleted due to unknown reasons or whatever the reasons, so that will leads to a specific diseases, and this could be tackled, like this could be studied, the pattern of this disorder could be studied to the pedigree by using what? Mendelian's principle. Hence we named it as, scientists named them as Mendelian's disorder. Clear? So this was all about the Mendelian's disorder. So I hope you still remember the task or the challenge, that is, we need to solve 50 MCQs per day. So I hope you are very serious with the task and you are also solving 50 MCQs per day, especially regarding biology. This is one point. So for that, I prefer you to, go with the MemoNeet app, as there are more than 15,000 questions framed based on the NTA pattern. This is one point. So previous year questions are also really very important. So you can also prefer the NTA PYQ book of the MemoNeet. And regarding any queries, and if you feel any difficulty regarding the present topic, you can reach me out easily through the message, or else even you can contact me through the Telegram. So the link is in the description. Make use of it, and thank you.